enzymes come in. Enzymes are special proteins whose job is to speed up the reactions. They behave differently from the structural proteins like those in your hair or nails. Instead of forming shapes in your body, they make chemistry happen faster. And that is why these enzymes are called the biological catalyst. A catalyst is something that helps a reaction to go faster without being used up. One way to imagine a busy road with a heavy traffic. The vehicles could still pass eventually, but the traffic would be slow and messy. But when a traffic officer steps in, the cars move smoothly and quickly. But the officer doesn't disappear in this process. In the same way, enzymes makes the reaction faster. Different enzymes have different jobs. Some enzymes break the large substrate into small pieces. Others join the substrate together to make something bigger, while some simply rearrange the molecule into the new form. So, how do these proteins actually do all of this? So, inside a cell, the enzyme molecules and substrate molecules are constantly moving. Every now and then they bump into one another with the right amount of energy and from the right angle. These enzymes are very specific in their action, but how they manage to do so with so many substrates around. So let's get into this lock and key hypothesis, which was proposed by Emil Fischer in 1894. He proposed that the enzymes work like locks and the substrates work like keys. The way a correct key fits neatly into a keyhole to open a specific lock, similarly, the substrate, when binds to a specific enzyme, only then the reaction can occur. Let's understand this hypothesis elaborately. Each enzyme has a special pocket where the substrate binds, and it is called the active site. According to the lock and key hypothesis, this active site has a fixed and a rigid shape. For the reaction to take place, the substrate must have a matching shape to these active sites. Only then a correct substrate can fit neatly into the enzyme. And this explains the enzyme specificity by forming an enzyme substrate complex. Then the reaction takes place inside these pockets. Finally, the products are released and the enzyme becomes free to find another substrate. This explains why enzymes are so specific. Like zymases, which act upon glucose to produce alcohol and carbon dioxide. In a similar way, we have a lot of enzymes which are working in our body, like carbohydrases, which only act on the carbohydrates, proteases, which only act on the proteins, and lipases, which break down fats into fatty acids and glycerol. Whether it is assembling or dismantling, the underlying idea is the same. Just like a lock and key, where the key completely fits into the keyhole, in the similar way, substrate fits into the active site, reaction occurs, and the enzyme comes out unchanged and ready for the new task. Scientists later developed more advanced models of enzyme action, but lock and key model remains starting point for understanding how enzymes work. So let's take up the takeaways from this lesson. Enzymes are catalytic proteins. Enzymes bind to specific substrates using a lock and key fit. Substrates bind at the active site of an enzyme. Active sites are complementary to the substrate. After catalysis, products are released and enzymes can then be reused again.
Molecules that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, is the area where enzyme activity takes place. An enzyme works by binding to molecules called substrates, at its active site. The job of the enzyme is to convert the substrate into a different product or products through a series of chemical reactions. Following the reactions, the products are released and the enzyme is free to act on another substrate. At the active site, the substrate should fit into the enzyme almost like a key fits into a lock. Because of this, most enzymes can fit only one substrate. To accomplish this lock and key fit, the active site undergoes a slight change in shape in order to better accommodate the substrate. This is called the induced fit model because the enzyme is induced to undergo small changes so that the substrate can achieve optimum fit. All enzymes have optimal environmental conditions that are correlated with maximum enzyme activity. Environmental conditions such as pH and temperature play a role in how efficient the enzyme is in conducting its reactions. When conditions are less than optimal, an enzyme will lose its configuration and slow its activity. This is due to a change in the three-dimensional shape of the enzyme and is called denaturation.